to this computer. There we go. Okay. I just keep these like on YouTube for the faculty to evaluate me, and just know that I'm actually here teaching, not just like in School of Rock when I'm like, all right, guys, don't do anything. See ya. All right. So red blood cells and white blood cells. The role of red blood cells: carry oxygen. That's it, pretty much. So this big molecule here in blue, that's hemoglobin. That's how it does it. So hemoglobin holds on to iron. Actually, it's going to be normally a 2+. We'll see what happens when it's 3. So Fe, ferric, that's iron. And that's pretty much the role of a red blood cell, is to use iron to grab oxygen and take it places. So you can kind of see the little iron molecules in blue being bound by the goofy hemoglobin, and then the oxygen molecules here in yellow getting grabbed and like basically distributed. This is the way that a large organism like us or other animals can distribute so much blood and oxygen and still feel pretty good about everything. So we'll take a look at a couple conditions where this hemoglobin is kind of out of commission and it's not as good. And that's where you can get something like anemia, for example, where you don't have enough red blood cells, you don't have enough oxygen running around, it's a bad time. Okay, the next system that we're gonna look at is the immune system. This is your white blood cells. So these are the two things that we're gonna look at today. And again, this is green text because I'm not gonna be testing you on the vast array of immune stuff. It's mainly there to kind of showcase what, what some of these cells we're gonna see today, what they can do. This is a GIF usually, sorry. But this immune cell, this is a big T cell, and it's just like eating this cancer cell. It's super cool. But yeah, I haven't figured out how to do GIFs on PDF. I don't think you can. Sorry. And sorry for the crowd that calls it GIF. That's been outvoted every time I've asked a class. All right, so some of you have seen this in lecture. So I'll just take like a formative assessment here in physical in-person lab. How much blood type stuff have you seen in lecture? Middle for like, yeah, it's kind of bad for like, we have not seen enough, and thumbs up like, we're pretty good at this already. Kind of like in the middle here. Okay, that's definitely the assessment that I got from the last class too. So this will be a good chance to kind of review everything and see kind of what happens. So there's one gene that has 2 different types. Well, 3 actually, as you can see right here. And then there's another gene, and that's the one that's the positive and negative gene. So put that in the back of your mind for later and focus instead on the A type, the B type, and essentially the 0 type, the O. So O and 0 can be kind of interchanged in this case. Okay, so as one of your in-house geneticists, it's my job to tell you, obviously, you have a pair of each chromosome. Humans have 23, so in total you have 46. One set of 23 came from mom, one from dad. So you have 2 shots at every gene. The blood type gene, the AB0 gene, is no different. You have 2 shots each time. So if you got, for example, 2 copies of the A, you're going to showcase on your red blood cell basically a piece of that A gene. It shows up as this big, like, massive receptor type thing. They have roles, but you don't have to worry about those for the class. So not only if you get 2 types of A, 2 sets from one from mom and one from dad, for example, you get the A blood type, or you still have the same exact A blood type if you only have 1. That's totally okay if you only have 1 copy, and instead you have that O copy right here. I know it's noted as I, but can you imagine doing a Punnett square with O's? It'd be really terrible. All right, same thing is gonna happen with the B subtype gene, okay? You can get 1 from mom, 1 from dad, and you can have the B type of receptor on there, or you only need 1 copy, get the whole package just with that. You're totally good. So there are 2 combinations with this gene that are unique though, past what we've seen. If you have, for example, the copy of A from your mom, the copy of B from your dad, you will express both types of receptor. This is your blood type in this case, in case you haven't. Okay, I think, yeah, because we were middle following, so we're good still. So if you are AB, it means that your red blood cells come complete with not one, but both in this case. And then there's the last option where both from mom and from dad, you were passed down the non-functioning gene, which has no receptors and it's a blank slate. Not necessarily a bad thing, as we'll see. But in this case, there's just nothing here. So if you have this type of gene, this is where you're type O, which is basically you have both versions of the gene that are just not going to express. Okay. Oh no, I did too much. I drew on the last lab and I can't erase this one from this app. So let's go here. Okay. Shout out some blood types. Who knows their blood type? O positive, okay. So there's one. B positive. And then we'll do me, AB negative. Actually, that's going to be really hard if we do that one. Give me another one, actually. Yeah. A positive. Okay. Let's do A negative just instead, because so that we have a negative in here. That's okay, though. All right. So in each case, the presence of the A gene is going to cause those little pieces to be on the outside of the red blood cell. Same thing for the B gene, but they're going to be B pieces on the outside. And nothing's going to be on the O type. Now the positive is a totally different gene. It's just positive, negative. It's either there or it's not. And that is called the RH for rhesus monkey gene. So that's where we first discovered it and we found out, oh, look, we share this. This is perfect. Okay. So let's take a look at the A subtype right here. This A subtype is going to have, let's call this in a matching blue. It's going to have those little blue pieces on it. Okay, we're good. It does not have anything else, as you can tell, because it's negative for the RH subtype, and it also doesn't have a B. So you only need to account for 3 things each time. So for the B positive, let's mark B's in red over here. It has the nice B antigens that are all put up here. And we'll learn a little bit about antigens and why we use that same name. Similarly, it has a positive. So this positive is similar to the A and the B's. It's just a totally different gene. It doesn't have the same variations. It's just on or off. But anytime you have a positive, you have the little positive cell surface things on your red blood cells. Okay. Same thing over here. O positive, you have the little positive RH gene. You're good. But that's it for the O positive in that case. O means no B's, no A's. That's it. So let's take a look here. Hey, this actually might be kind of hard for everybody. Okay, let's do this one. This will be easy. All right. Can this B positive person, let's say in an emergency, transfer their blood to the O positive person? And so again, you don't have to answer, but think to yourself, about 8 seconds. And remember kind of a golden rule about transfusions. Whoever you're sending that blood into had better seen that type in their blood before. So if we break this down piece by piece, sure, this has that positive gene right here. So does this B positive, good. But this person has B genes, and this person doesn't. That's not going to be a good deal. So what happens is this is actually a red blood cell, white blood cell crossover a little bit here, is that if you send in this B positive gene with all these little B pieces right here, the immune system of this O positive person thinks that that is a pathogen. It thinks it's some sort of virus, bacteria that's coming to kill the body. Your immune system is super powerful and it's super angry all the time. So if you send in donor blood to a recipient that does not have or recognize that B antigen, that B receptor up there, they'll go into what we call graft versus host disease. And their entire immune system basically nukes their entire body, thinking that they're under some massive infection because you put the wrong blood in them. This was the sad fate for so many rabbits as we tried to perfect transfusions and putting blood into rabbit A from rabbit B, and they kept dying. We didn't have this set up yet, unfortunately. So poor rabbits in that case for sure. So let's change this up a little bit though and go the opposite direction. Now the O positive person is giving their blood to the B positive person. does the B positive person and does their immune system recognize everything that the O positive person is throwing at them, right? The O positive person is only throwing these green antigens from the positive Rh gene, rhesus. The B positive person has seen those before. We're doing pretty good. Okay. So keep that in mind. The best donors are those that have very few of these little receptors on their surface. The best acceptors are those that have the immune system that has everything already complete, basically. So I'll let you think of this one too. Would this person be able to transfer into here, for example? This is just kind of your last thought experiment. So yeah, even if you send in, even though this person does not have the Rh gene, if you try and send these in to a bloodstream that does not recognize the A antigen, everything goes haywire. You're in a lot of trouble again. So blood type is a very good intro for all the kinds of stuff that you have to manage when you are treating a patient or a client in some cases, depending on your PT or EP job. You have to really make sure whatever you're giving the patient is going to match up. Blood type's a good intro to that because when we calculate really what you can give patients or transplants specifically, it's like hundreds of different things. Luckily, we have machines that can tell us a few of those things, but it does not hurt to have a human in the room, right? You wouldn't want to be fully treated by a machine, right? Okay, so we can clear that. Cool. All right, so let's actually go to your actual lab notebook, and you can take notes alongside here if you want, but again, I'm probably just gonna make like little like stars and stuff for stuff to raise your attention to. So again, with your notebook, what you'll have is a complete set of information. Don't let this, again, lull you into thinking like, I've read it once, we're good, it makes sense, move on. Make sure that you can retrieval practice this, put it on a blank page and say exactly why an AB positive person can take any blood, for example. Come to me if any of the terminology ever gets weird. So remember all the receptors and those little colorful things that we just called, we call those antigens. You can also call them receptors, genes, anything you want. And let's say on a test, if I call it an antigen and you're like, is this the same thing as a receptor? Just let me know. And we can, that's an easy clarification. Okay. I know. So anytime you look at antibodies though, so antibodies are your immune system's reaction to things and what they know is available. Antibodies basically are produced when something, when something shows up that's okay, or that's not good actually. So anytime you have an antibody show up, it shows up against something that is not usually present in the body. So somebody with A, they have A antigens, so they're good. But if you put B positive blood in them, you're going to force the immune system to make antibodies against B positive blood. And that'll rip things up in the patient. It's not good if you transfuse blood into them. So that's how you can use both of these tables to kind of check, okay, what am I looking at here? Here's that Rh version. It's literally just on or it's off. That's it. A lot easier. It is still something that you want to make sure you're combining in each case because they always come together. See if I can go like that. So your full blood types will be the presence of A, B, both, and then the presence positive or negative of the Rh antigen, which is just the positive minus. So there's some really good stuff in here if you plan to be an OBGYN about how the fetus, if it has a different blood type than the mother, you have to really watch out because different blood types don't mix very well. And they can cause a bunch of anger in the mom's immune system, in the baby's immune system and stuff like that. Penny did a really good job kind of talking about how difficult it is to catch. But once you catch it, it's okay because you can actually tell the mom, you can give her immune system a blueprint not to hurt the baby, even though it looks like it's a foreign pathogen in that case. But I won't elaborate too much on that. It's really good reading though, if you want. Okay. Oh, look, we're already done. Hooray. That was quick, right? Okay, so let's go here and then on to 12. Okay. All right. So 12 is the main one, I'd say. 12 is the bigger one. So now we're more in the realm. We're still going to cover a couple things about red blood cells just so you're prepared for like kind of what diseases red blood cells can have. But the transfusions and who can take what, that's easily the biggest set of questions that we can put on the test, right? Like you have a patient roll in, they're B negative, give me the bloods that they can have, done. And that is a decision you do have to make fast sometimes. So I am a B-cell immunologist by research training, so I kind of did too much in the last section, so you're going to get the light version, and we're going to move on because it's Wednesday, and I also have a meeting at 2, like some of you have said too, so that's okay. Anyway, though, cool thing about white blood cells, these things are massively powerful. Our body fights all kinds of stuff all the time. Again, a GIF that I don't have working, sorry. This is a massive parasitic worm. It is surrounded like a wolf pack by your white blood cells, and they attack it, and they basically burn it to pieces. And it's super coordinated and super devastating. So it's awesome that we have this in our bodies, and it takes care of us all the time. And I'm a cancer person too, and one of the main hypotheses behind cancer is something called surveillance hypothesis, which is cancer arises in your body thousands of times throughout your life, but your immune system catches it almost all the time. That's how powerful it's theorized to be, at least, or hypothesized to be. The other thing is that we don't like stuff like this inside of us, obviously, and we need something that can go in and like really get it. Okay, so this is also a picture of GVHD. This is when you put the wrong blood type in somebody. It's just like a big, wide systemic inflammation, basically excite the entire immune system. So this is actually a good example to note that power that you had against the parasites, against bacteria, viruses, that power can be turned against you too. That's autoimmunity. That's when somebody's immune system is too powerful for their own good, or gets targeted and pointed in the wrong direction. So this is the consequence that you look at, is turning that your own body's basically your own body's super weapon against you, and it'll just rip things apart, and it's not a good time. Okay. This is neat, we can talk about this later, but our immune system is kind of strange compared to other animals because as Homo sapiens evolved, we did a few things differently, include keeping lots of animals in close proximity. That was kind of the birth of most of our modern diseases, smallpox, malaria, all kinds of bad stuff. So thanks, livestock, right? Sorry I annotated this, I should have reset it. Okay. Last one on the white blood cell stuff, and again, a green text, but those of you in healthcare, you're gonna keep seeing these characters a little bit. Some of these are more of a threat than others. Obviously, we're not doing a great job on viruses still. Highly adaptable, quick, hidden, hides in the DNA, super bad. Oops. Prokaryotes are usually pretty specialized, or no, sorry, protozoa. Prokaryotes are bacteria. That's a bacterial infection. They have different traits and advantages that viruses don't. They're a lot more like a living thing. They're a little more adaptable. Fungi can't get into humans most of the time because we run so hot, but these are devastating to amphibians, plants, agriculture, things like that. And there are a few that are starting to live inside humans. It's not a good deal. Protozoa, this is your malaria, things like that, but it's also amoeba that eat your brain because your brain looks like their normal food. So I'm not kidding. Next time you're at the lake, don't take any up your nose, any of the water. No joke, I'm not kidding. So pretty bad. Lastly, prions. Prions are basically just a misfolded gene of yours that clumps up in your brain and takes up room. No more brain room, no more action potential. It's a bad time. There's no cure and there's nothing your immune system can do. But the cool thing that we've actually conquered in the last, say, 100 years is parasites. Our immune system is super directed against parasites. But today we rarely encounter parasites because of a lot of public health changes in a lot of cases. Washing hands, filtering water, things like that. So those steps can usually eliminate a lot of parasites, not all of them, unfortunately. But you still have a lot of immune cells that are super focused on this. And we'll see a couple of those today, actually. Okay. Yeah, we're not gonna do that. Okay. If you guys ever want to talk about immunology, let me know, and like we'll totally talk about it. So, but I did want to give you a good like reference for what's going to be on the test, things like that. Okay. Hemocrit is just when you take somebody's blood and you spin it down super fast, like in a gravitron at the fair, and you get kind of a measurement of how many red blood cells do you get, and that's going to be in red down here. How many white blood cells did you get? That's plasma. If you have too many white blood cells, that's a sign of infection, because remember, white blood cells are your immune system fighting something. So just the off and on presence of white blood cells is usually a pretty good sign that something's going wrong, or that you have some sort of chronic immune issue that is taking up your time. Okay. So you get to see a couple of these in the slides today, platelets. So erythrocytes, that's just red blood cell. Don't worry about that. Platelets are these little chunks of clotting material. So they run throughout your blood, and if they detect a cut or an opening or some injury, they start collecting together. They start forming big structures, and that's the clot that you see. So like I cut myself peeling potatoes because I'm a moron and I peeled myself. And so everything clotted because all the platelets basically found like, uh-oh, there's an opening here. And they usually detect oxygen from the environment and they're like, oh no, and they clog that section because you don't want anything from the environment getting in your body. Your skin's one of your best immune system like barriers, basically. So this goes into a lot greater detail right here about hemoglobin and the issues that can arise because of it. So sometimes you get, let's just do highlighter. Sometimes you get hemoglobin in the wrong form. And in this case, it's actually going to be Fe3. It's a small detail. I'm not going to test you on that. It would be more of an example of like, which of these following diseases have to do with red blood cells, not white blood cells. And I would select something like that. Just know that red blood cells are differentiated from the white ones, that they have hemoglobin. And when stuff goes wrong with hemoglobin, you're talking red blood cell stuff. Okay. Last one, and this is super nasty for red blood cells, carboxyhemoglobin. Let's get rid of that. This is when carbon monoxide shows up, and carbon monoxide is way better chemically at binding your red blood cells than oxygen is, and it takes oxygen's spot. Just kicks it right off. So instead of distributing oxygen to your brain, you're now distributing CO. And that doesn't even go into the fact that carbon monoxide also is going to poison all your mitochondria and all your cells. That's not a good time either. So that's why carbon monoxide can act so quickly. And you also can't smell it, anything like that. So obviously get like one of the alert things for your house. Okay. So let's take a look at some of the leukocytes. This section right here. So if you're not on this page, get to this page now, because this is I'm going to tell you exactly what you're looking at in the microscope slides for that activity, and just take some notes on what you're going to see in this case. So this is going to introduce you to a few white blood cell characters. Remember, like I said, your immune system has to be pretty vast to take care of all kinds of different stuff that's trying to kill you. Different things have different specialities too. I know I say that word wrong. Okay, so let's take a look at eosinophils. Let me know if you find one of these. They are super rare in the slides. They're red. Most of the things that we're going to stain today are going to show up purple. So you can put a little note on that. Is that anything that you see today? Let's see if I can change this one. Yeah, I totally can. The stain that we're going to see today is what we throw at these cells to see, okay, what kind of cells are they? This stain binds DNA. It's like ink, and it grabs DNA and it marks it. And it's also going to be in purple, except if it's an eosinophil, it's going to be in red, strangely. Okay, that's the main driving characteristic of an eosinophil. The reason that they're rare is that they fight parasites. We don't have parasites really in our bodies anymore, chronically like we used to. Homo sapiens were not much different than any other animal, and we'd do all kinds of stuff to get parasites in us. And so our immune system was constantly fighting those things. So they're our biggest enemy that we used to have. We don't have them anymore. But it's kind of like in Jurassic World when Chris Pratt's like, okay, this thing woke up in this century. It has no clue that parasites are solved anymore, but it's still super angry. So people with super bad autoimmunity or allergic reactions, they have a ton of eosinophils. Basically, they think parasites are around when they're not, and they go off killing stuff. So that's kind of a chronic issue for eosinophils when they're super high. So I think I can write. So the main thing here is that these ones are red. Let's go here a little bit. But they should have this kind of nucleus too. Okay. So when you're staining, look for these shapes. like right here, and then we'll see these ones right here too. So basophils, kind of the same thing as eosinophils. They're going to have this same shape too. Again, these are one of your cells that your body doesn't really use anymore, but they're still really angry and looking for parasites to kill. So they usually take that out on plant pollen, unfortunately. So when plant pollen gets in you, your body thinks that it is some sort of foreign invader and it goes nuts trying to kill it. It usually thinks they're a parasite because a plant pollen looks like a eukaryotic cell. That's what parasites are. So both of these are actually really good examples of autoimmunity. So I'll just put that as AI. All right. So let's move on to a couple of cells that you will meet and your patients will meet, and they're a very good sign of something's wrong or that an infection is at least being cleared in the moment. So you're used to seeing these guys all around. Neutrophils, and they are this type of cell up here. They have this goofy, like segmented, like chunked nucleus here. You'll see this structure in neutrophils. So this marks itself as a neutrophil if you see that goofy, like piece by piece purple stain. So neutrophils and actually everything on this page can be considered part of your non-specific immune system. This means that it's not targeted. They just exist and they kill stuff that looks wrong. That's it. So neutrophils are your kind of your frontline, I guess, soldiers in this case. So if you cut your hand, bacteria will flow in because bacteria are all over your hand. It's just, even if you wash your hands all the time. When bacteria show up, neutrophils get activated. You do not want neutrophils activated if there's not infection because they're very powerful. So all these little granules, you see that marked here, all these little granules right here, and the reason they actually have a role, and you'll see them in the microscope. What these do when bacteria show up is they basically shed out and explode, and they're full of poison, and they kill bacteria. So they do massive amounts of damage, neutrophils do, because they are the frontline response. They're the thing that stops bacterial infections before they can take hold. Because remember, bacteria can multiply exponentially. You gotta really get this quickly. And neutrophils are your massive like nuke that you send in to just completely blank everything. So when you have high neutrophils, this is kind of more of a clinical example. High neutrophils in a patient is showing, okay, this person either has a chronic infection or an acute infection right now, but if they have chronically high neutrophils, they shouldn't be turned on that high all the time. And that's when you have to look into like, okay, what's causing this infection to keep these neutrophils going? Because you don't want that in the patient, it's too much. All right, so eosinophils, basophils are this one right here. Neutrophils, they're this one. So you'll do those counts today. Here is a monocyte. And most of the time, monocytes, you can also call these macrophages. That's not on the test, so I'll call them monocytes. What these do is they show up to sites of infection and they start eating the bacteria. That's what all this room is for. Yum, gross. In this case, you'll see this big, giant, kind of kidney-shaped thing. And these are typically going to be bigger. And the other main characteristic is that they have to have all this room right here to eat the bacteria. So look for a big empty space within the kind of within that cell circle too. These are a sign of a bit more advanced infection than neutrophils. If neutrophils can't handle it, these show up. So you can kind of think of these as a second wave. Think of these as more of a first wave. And zero wave is stuff like your skin and like mucous membranes that just catch stuff before it ever gets in. That's not on the test, but intuitively, I think that helps sometimes. All right, cool. So we met a lot of these cells. And I actually might send this video to the last section because I tried to do this more like, look what happens here, and I didn't actually just do it on the notes. And so if you're here from section 5, welcome to the actual stuff that's on the test. Sorry, I got excited. I'm an immunologist after all. All right, last couple people we're gonna meet, or last couple things. Lymphocytes, there's 2 types, but you're only responsible for finding this structure to see if it's a lymphocyte. Okay. In this case, these are gonna kind of look just like a big full moon, all shaded, all dark, nice and even, no shapes. Okay. Is your third and final wave against an infection. And they're good, it's a good intro for this, so take about 8-10 seconds and think. And if you have read through this already and kind of previewed these notes or remembered what Penny talked about with B and T cells, why does it take a patient about 3 to 7 days to get over a sickness when it's really bad, right? Your first and second wave failed, or at least they're fighting as hard as they can, but it's not winning. This is the final thing that you need to do to eliminate something. Everything before lymphocytes that we covered was something we call non-specific immunity. It's just natural killer cells that show up and they kill stuff that looks foreign, bacteria, viruses, parasites. Sometimes you need something a little more specific. It's actually a key COVID aspect between who's asymptomatic and who's not. Those of you that are asymptomatic may have defeated the virus in that first non-specific immunity, and there's certain genes and physiological differences between who has that non-specific immunity versus who has to go through this process in those 3 to 7 days. And the reasoning behind the 3 to 7 days is that what B and T cells do is that they evolve inside your body in real time to create molecules that bind perfectly to whatever has showed up in this time. This is well thought out by this green text right here. How is it possible that you could encounter something that no human's ever seen before, some sort of pathogen, but your immune system does have the tools somehow to come up with some response? And for a long time, people didn't have a good answer for this. And I hate that this example has Giardia because I was like tubing in the Colorado River when I was growing up, and I got Giardia once. That was a bad time. So stay away from the water, everybody. It's not as fun as you think. Brain-eating amoebas, Giardia, it's awful, gross. Just stay safe in your house and read, right? That's what all professors should tell everybody, right? This is the infinity problem, though, that anything could be a pathogen that nobody's ever seen before. It's your B and T cells that do that. So what B and T cells do is they break off chunks of whatever you're looking for. They take that chunk and they design themselves to find it. So they take a piece off of a virus, a new virus or new bacteria. And again, that's called an antigen. So remember back to red blood cells. If your immune system has never seen the B antigen before, it thinks it's a pathogen it's never seen, we're in deep trouble. Starts freaking out. Oops, there we go. Oh, we're not gonna do that. We're not gonna do that, sorry. Okay. See, I am gonna try and take it a little easier than I did in that first one. Okay, so whenever you see one of these things, and let's say you see a big enrichment of these lymphocytes, it means that you do have an advanced infection on your hands because these are the final wave. And what they do is basically make large targets out of themselves to go find whatever's killing you. Well, hunting you. I shouldn't say it's already done it. So let's go here. Okay. I know the eraser drives my OCD nuts if I can't get all the little pieces. I don't know, so terrible. So if we have a B cell here, for example, what these do is actually have little Y-shaped molecules on their surface called antibodies. So these antibodies can go and bind, let's say this is a piece of virus or something. What they do is they recognize that piece of virus and they're like, oh, look, we can recognize this. let's make way more of me. So once you get a B cell in your big pool of evolving B cells, which you constantly have, and it's constantly actually mutating your genome to make better B cells, everybody has this. You find the one that can actually bind it, you make tons of them, and then you engineer, and then you start secreting the antibodies out into your bloodstream, and they essentially go and bind and kill whatever's been basically attacking you for 3 to 7 days, and you can kind of get over it. T cells are a little different. They actually go and hunt and kill the specific cells, but again, they go through kind of a hyper-evolution to make sure that they are ready to find and target whatever is offending in that case. So again, a lot of this stuff, a lot of the backgrounds in here, it's very good. Both B and T cells, again, can trigger autoimmunity. For example, your B cells, if you have type 1 diabetes, your B cells have treated your pancreatic cells as an enemy for whatever reason. They've mistargeted it, and they're going to keep targeting those cells, unfortunately. We're working on certain ways to combat that, but that'll be good. Okay, so you do not need to do the microscope here. Use the links to play the blood game. Use the links to go to the tissue slide and zoom in on it. That's actually how most tissue slides are evaluated, is not like in person live. Let me know if you really want to, and we can get you scrolling on this microscope, but for the sake of your Zoom partner too, it's probably best if you do it online and share the screen. It's a lot clearer of a picture too. So the blood typing game is pretty easy. That's pretty much just saying like, okay, like what type can you give this person? What happens if you give them the wrong one, etc. Then doing the count is exercise 12. You got to find all of these things, count them, at least 50 total of the white blood cells, not 50 neutrophils, not 50 eosinophils, things like that. Just find 50 total white blood cells. One last note, and you can just put it down here. When you're looking at a blood slide, you're going to see a lot of little like empty, clear cells. Those are red blood cells. Red blood cells kick out their DNA to make room for more hemoglobin. So they're just going to show up as just a blank slate. You're only looking for stuff that looks like really dense and dark, like these nuclei are going to. Okay. All right, so I think we're pretty good, right? Did everybody feel okay? Pretty good? Not too bad, hopefully. But yeah, so here you can just do like, okay, I found like 5 of these, only 1 of those, 3 of these, and just like so on and so forth. Using this and counting clinical slides and pathology slides is a very good and cheap and effective way to understand where somebody's at in their cancer diagnosis, in their infection prognosis, in their autoimmunity. And it's a whole lot better than crazy imaging and tests that otherwise you'd have to pay for, or well, the patient has to pay for, and thus we all have to pay for. Okay, let's do, I think we're all set. Yeah, so give me a sec on the Zoom rooms. I'll get everybody in. First things first, if you're on the, if you're in person, log on to the Zoom so your partner can join. Next, do the blood typing game. It is an Adobe Flash Player game. That's dying in December, unfortunately. Basically because Flash, Adobe is a really easy way to get malware into things. That's what I've heard. I'm really sad about it. You have to kind of hit some buttons in Chrome or Firefox or whatever Internet Explorer is now to basically get everything working. You just have to allow it and like basically say this pop-up is allowed to work on my computer. And then the tissue slide stuff, that's another link that's on D2L again. Or I think you can search it from your lab notebook and just Google it. Just search tissue slides, search 103, that's our patient 103, and then start tallying what white blood cells you have. So I think we're all set. Everybody feel good? Flag me down if there's anything I can help with, and we should be good. Okay.